Good afternoon to you all. It is a pleasure to be here. And before I start, I would like to thank Land and its organizers for inviting me, especially Emily Murray for making the first contact and for allowing me to be here sharing my experience. And I also want to applaud this initiative of organizing these very important and interesting seminars. So, as team presented, I am a researcher at the Public Health Institute, University of Porto. I work on the health impact of neighborhood environments, and I am also an invited assistant professor at the Faculty of Medicine, also in the University of Porto. And today I'm going to speak about the geospatial data that we have available within this very large bird cohort, Generation 21 bird cohort. And I'm also going to talk about the use of this geospatial information for a specific project that I coordinated during the past 4 years entitled EXALAR project. So for those who do not know the place where I work, which I believe is most of you, because Porto is a small town, we are not very well known in the field. So this is the building where I work. I work in the office right on the top floor, in the fourth floor. This building from the Public Health Institute is located right in the city center of Porto, in what we call the Old Town, so the historical center. So every day we hire about 50 people here permanently, plus graduated students. And the people that work in the institute, most of them conduct research within one of these cohorts that you can see in this timeline. So since 1999, with the creation and establishment of the first population-based cohort in Portugal called EpiPorto, which is a cohort of adults. Since then, several other cohorts have been recruited and followed up. Some of these cohorts are population-based cohorts, such as the EpiPorto, the EpiTeen, and the Generation 21, which mean they want to be representative of the population that lives in Porto metropolitan area, while others are what we call specialized cohorts. So they focus on subgroups of population, such as drug users, preterm babies, homosexual men, twins, cancer survivors, etc. In this presentation, I'm going to speak about Generation 21, which for those who do not know, is actually one of the largest birth cohorts in Europe. These cohorts included all live newborns born in 5 main maternity hospitals in Porto metropolitan area, and it comprises about 8,600 newborns that have been followed up since April 2005. And so far, we had 5 evaluations of the cohort, full evaluations. So all members of the cohort have been evaluated at 0, so recruitment, 4, 7, 10, and 13. We also have a couple of evaluations that only included a subsample of the cohort, which I'm not going to speak about. And we are at the moment organizing the 17 years old evaluation. We did the 15 years old evaluation because of the pandemic, so we started it, but it was stopped in the middle of the evaluation. This cohort was designed and conceptualized by Professor Henrique Barros, which is the director of the Public Health Institute, and it is coordinated by Professor Ana Cristina Santos. So if you want more information and about the cohort, you can always get in touch with these 2 important people. And you can also visit the official website, Generation 21, as well as the, I think the website is birdcohorts.net or birdcohortseurope.net, where Generation 21 also has page feature there. So we know a lot about these children, which are now about 16 years old, and we also know a lot about their parents and their families, more precisely their mother and father. Some of the information in the cohort is retrieved by questionnaires that are applied during interviews and that allow us to know a wide range of issues such as socioeconomic characteristics, eating habits, physical activity, diseases, violence, bullying, sleep behaviors, etc. I'm not going to list all the variables that are available because it would be too much for this talk, but if you have any particular question, you can also ask me at the end. We also make a big effort to collect more objective measures of health during the physical examinations conducted at each evaluation. So since birth, we have a lot of information with these regards. We have anthropometric measurements, body composition measures. We have all kinds of indicators of respiratory and cardiovascular function, bone densitometry, and also a pretty complete range of biochemical parameters that are assessed from blood samples that are collected at each evaluation of the cohort. Besides, it is also important to state that we have a quite large biobank. So we store biological samples about these children and about their parents in some instance. More precisely, we store total blood, we store serum. At birth, we store umbilical cord, total blood. We also have the baby tooths collected, and we have extracted DNA that allow us to measure things such as DNA methylation. So this biobank is quite important because it allows us to go back in time and to keep the cohort alive by adding further information on biomarkers that are relevant for specific projects and also for specific time points. So for instance, as I will mention in the EXALAR project, we retrieved serum samples collected at 10 years, and we conducted a series of tests to see if these children had some kind of allergy. So this biobank was particularly important and valuable for the project. So as you may already understood so far in this presentation, and contrasting with cohorts that you might be more familiar and that you conduct your research with, in this cohort and in Porto cohorts, we do not ask participants about their neighborhood environment. So we don't have subjective assessments on the quality of the neighborhoods of living. We don't know what's the participant perception on the availability of green space, on the quantity of pollution and noise. So we don't have that kind of subjective information. And this happens because this cohort was at the inception designed to study cardiometabolic health and the biological and individual level determinants of cardiometabolic health, as well as predictors of adverse perinatal outcomes. However, it is also important to mention that the city of Porto, the metropolitan area where it stays, and Portugal as a whole, is a quite heterogeneous country. And we have wide gaps in terms of life expectancy between regions and between municipalities. And this apparently seems to be related with and also an equal distribution of the material resources, of the healthcare services, and also of the environmental resources available to the population. Therefore, and also seeing the increasing evidence coming from mostly studies in the US and the UK about the role of neighborhoods and on health, we also started to develop research in this field and address how places where individuals live affect their physical and mental health. So in Generation 21, as I mentioned, we do not ask directly participants about their geographical context of living, but we ask them their full residential address, and we also ask for the name and location of the schools they attend. So we are able to identify the coordinates of these locations and to produce maps and exposure variables that I'm going to speak about. So in this map, you can see, just to contextualize, in your left-hand side, you can see that dark area. This corresponds to the Porto metropolitan area. So the children from these cohorts are supposed to be representative of the population from these areas, quite a big chunk. It holds about 20%, or not 20, 15% of the Portuguese population. So it has a lot of population living there. In the map from the middle, you can see the participants' residential locations at birth. So since then, there has been quite a dispersion around this core area because these families move houses a lot. And in the map C, you can see the school locations at 7 years of age when children were already attending primary schools. So a bit of technicality. So how do we manage this address information? So at each assessment, participants and their caregivers are asked to provide their full address. These full address are then verified and clean, and then they are georeferenced or geocode as you prefer. So you use a combination of automatic and manual methods of geocoding. So we start with the automatic one. So, and for that we use ArcGIS online geocoding service that uses a street dataset, the dataset from the company EAR, is a navigation company. And we also use Google Maps geocoding API that uses TeleAtlas as database. For approximately 20% of the address, we cannot use these automatic strategies, so we use manual ones. In some cases, we ask the CTOs and the local governments to provide us up-to-date street cartography so that we can pinpoint these locations and identify the coordinates. In some cases, namely in circumstances where we have residents in substandard housing, we need to go to the field and collect the locations with a GPS. So I don't know if you are aware, but in Porto we have a specific type of housing called ilhas. In English, it can be translated to islands, which are illegal forms of construction that emerged with the Industrial Revolution, which in Porto is quite late, and they still subsist in the urban fabric. And these kind of houses do not have official door numbers, so we need to enter these big gates and go through a quite narrow street and collect the locations of these houses using a GPS. It is also worth mentioning that we have implemented all necessary procedures to ensure the people's privacy and anonymity. So the geocoding procedures are done at the Public Health Institute by me and by students that work with me. So the service is not subcontracted. The procedures are conducted in desktop computers connected to the local network. We use anonymized data sets. Whenever we want to represent the locations of the participants in maps, such as the ones I show you, we use a location fuzzing mask, so we kind of misplace the points so that you cannot identify the true location. And whenever some researcher asks for information on exposures, environmental exposures assessed by linking the locations of the participants, we do not provide you coordinate data, only the exposure data. So we follow this workflow in terms of management. So we have the cohort database, and the GIS analyst is provided with the addresses without any kind of health data associated, and with an ID that is different from the ID original of the cohort. And only the database manager knows this correspondence between IDs. Then the GIS analyst conducts the geocoding procedures, the exposure assessment, and it sends back to the database manager the coordinates and the exposures, and the database manager establishes this correspondence. And then whenever someone wants to make a data request to the cohort, the person has to submit the project to the scientific committee, the ethics committee, and in the end, it will receive only health data and exposures without any kind of geocode or coordinates. Here you can see some stats on these procedures. Putting together schools and residential locations until the 10-year evaluation, we geocoded about 12,000 addresses with a match rate of 90%. And this was mostly done using automatic strategies. So after this quite time-consuming task, we can then assign neighborhood exposures to each participant's location finally. And here you can see a list of some of the variables that are available within the cohort. Again, I'm not going to be too much detail in this slide because I still need to talk about the EXALAR project, but we have quite a lot of information related with different topics and different exposures, things like walkability, pedestrian access to services and community resources, including green space, food environment characteristics, measures of neighborhood deprivation, etc. And since we have these coordinates and geocodes, we can link the cohort data to an infinite amount of environmental information already collected by other agencies and institutions. So moving to the second part of the talk, the EXALAR project. So this project took place between April 2018 and May 2021. And this project was funded by the National Science Foundation and aimed to study how the exposure to urban air pollutants, green and blue spaces, and biodiversity influence the development of allergic diseases and asthma in children. So if I'm just going to briefly go through the project, but if you want to get more details, you can always visit the official website and the corresponding Twitter page. First of all, I just want to give a bit of context. Why do we decided to conduct this study? I don't know if you are aware, some of you are parents, so you might be very much aware of this, but allergic diseases have a huge burden in pediatric population. It has been estimated that 1 out of 4 school-age children have allergies, and 50% of the European children have asthma. It is also important to state that these two diseases are very much connected. So children who have asthma also tend to have allergies at the same time. In addition, it was also a big motivation for us, the trends that we see nowadays regarding these two diseases. So allergic diseases and asthma are increasing in terms of prevalence all over the world. And there has been a huge reflection around the medical and scientific community on the reasons for this increase. And what is on the table at the moment is that these increasing incidence and prevalence in inflammatory and immune diseases, because asthma and allergic disease are inflammatory and immune diseases, can be related with urbanization and can be related with a reduction in the contacts with the biodiversity and nature. So this is what they call the biodiversity hypothesis, that states that reduced contact with a variety of fauna and flora species leads to a poorly developed immune system and therefore to abnormal responses to common allergens. So in the EXALAR project, we wanted to test these hypotheses using our Generation 21 datasets and the exposures in Porto metropolitan area. So as expected, we had to collect quite a lot of information on natural spaces, namely green space and blue space. So we use one of the most widely used metrics of greenness, the NDVY, and I'm not going into much detail because I believe most of you are already work with the index or are well aware of the way it can be interpreted. But the index ranges from -1 to 1, where values around 0 or below 0 mean bare land or water bodies, and values near 1 means that that particular area has a lot of healthy vegetation. This index, and in the case of the EXALAR project, was calculated using Landsat imagery, which have a good resolution for local studies with a resolution of 30 meters per 30 meters. And we calculated the average NDVY around participants' homes and schools at different buffers, 100-meter buffer, 250-meter buffer, and 500-meter buffer. And we did this for the evaluations from 0 until 10 years of age. So we have 4 assessments of these measures. Besides, we also determined geographical accessibility to urban green spaces because this measure was not very widely used in the literature on the topic. And also we believe that probably these spaces are more attractive to parents and caregivers, and therefore these children would benefit more, would be more likely to visit these spaces and benefit more from the resources, natural resources available there. So the maps and cartography of these urban green spaces was provided by city councils. Some of the city councils do not provide us the information because they believe it's confidential, and we had to go through listings and to develop our own cartography using satellite imagery. And in the end, based on this cartography and based on street data set, we determine whether children have these spaces in the vicinity of their homes and schools. We also measured the number of spaces as well as the shortest distance to them. And we considered vicinity distance until distance of 400 meters and 800 meters because they correspond roughly to 5 and 10 minutes walking distance, and they are considered appropriate walking distance for children. Besides, thanks to the kindness of the Porto City Hall, in addition to urban green spaces, we were also able to assess and the location of all the habitats in the city, which is what you can see in the map. And for each of these polygons that corresponds to habitats, we have the number of animal species that live there. So this cartography allows us to test in a more objective manner the biodiversity hypothesis that was under discussion, and that believes that having a lot of species around our homes and having a lot of contact with these species helps to develop a healthy immunity. We also consider access to blue spaces. Again, this was something that was poorly addressed in the literature, but blue spaces also offer opportunities of contact with nature, and therefore they may be implicated in the etiology of these 2 outcomes we assess in EXALAR project. So in Generation 21, we basically measure the accessibility to water bodies such as rivers, streams, lakes, and the sea. And we use for that cartography data from the Portuguese Water Atlas that is publicly available and is disseminated by the Portuguese Environmental Agency. And finally, air pollution. So air pollution was not our main focus on this project because of two main reasons. Firstly, it is probably the environmental exposure that is more explored in the literature, and also because we had a few technical, not few, quite a lot, technical problems in assessing air pollution in Porto metropolitan area. So we end up using air pollution as a mediator variable and not as a main exposure variable in this project. So in terms of the technical difficulties, I think it's important to share because you might have some solutions for our problem. So I don't know if you are aware, but Porto or any of the Portuguese cities were involved in the ESCAPE project, that big European project that produce local estimates of air pollution. So we don't have that kind of information. And in addition, we have a very sparse network of air quality monitoring stations. So as you can see from the map on your left-hand side, that's the only monitoring stations we have for the whole metropolitan area, which means that every time we try to model this, we end up with estimates with a lot of uncertainty. So we are still figuring out what is the best approach for our case. So in the EXALAR project, because of these technical problems and technical difficulties, we end up using the air-based interpolated surface of the produced by the European Environmental Agency, which have a resolution of 1 per 1 kilometers, which is not ideal, but well, that's what we could do. In terms of outcomes, so to determine the presence of asthma in these children, we use this well-known questionnaire, the EXACT questionnaire, which has several questions on symptoms, respiratory and allergic symptoms, and allow us to accurately classify the children as being asthmatic or not, and as having allergies or not. Besides, we also consider more objective assessments. So we included in this project lung function parameters that were assessed through spirometry at 10 years of age. And we included parameters related with airway obstruction and small airways narrowing. And to measure allergy, or at least the high probability of children being allergic to something, we use these antibody tests using the immunocap assay based on serum samples that we retrieved from our biobank. And we were able to create, well, to dichotomize the kids into those with a positive result. So those that had an allergic sensitization to one of or series of major allergens, and those who do not have any kind of sensitization. So moving to the results. So I just list a number of key findings from this project. There are more, but I think these are the ones that are more relevant to bring to you. So our first key finding was that, and this is the finding that is less consistent indeed, because we found different results depending on the buffer size we use. But overall, we found that children that lived in greener neighborhoods, as assessed by the NDVY, had lower odds of being asthmatic, regardless of the levels of species richness. So that variable on the amount of species in the neighborhood surroundings ended up not being that important, and indeed we found quite more harmful effects than beneficial effects. We can then discuss why the measure is not perfect, so it has a lot of problems. So this is probably the explanation for this finding. Our second main conclusion was that allergic sensitization, which was assessed using those serum samples at 10 years of age, was less common in children with higher late life. When I say late life, it means, well, 7-10 years of age. So bear in mind that the life course I'm considering is quite short. So children with higher late life exposure to greenness and high stable trajectories of greenness exposure throughout the life course were less likely to develop allergic sensitization. So you can see in the table the associations between NDVI, distance to blue spaces, and allergic sensitization at 10 years. And what we found was that exposure to higher levels of NDVI at 10 years was associated with a lower risk of developing allergic sensitization, while the associations observed for the earlier time points were not statistically significant at least. We also were able in this study to identify 4 trajectories of vegetation exposure from 0 until 10 years of age that you can see in this graph. So we have children that always had high levels of NDVI exposure through their life course, those who always had low levels of exposure. Some of them increase the amount of vegetation they have in the home surroundings, and some of them decrease their levels of exposure. And what we found was that comparing two children whose NDVI levels around the residence decreased through time, those with high stable levels of exposure had a significantly lower risk of developing allergic sensitization. Another finding of this study was one related with lung function that we assessed through spirometry at 10 years of age of these children. What we found was that lung function, more precisely parameters related with airway narrowing, was better among children with increasing levels of NDVI from 0 to 10 years of age. So in this table, you can see the life course metrics that we calculate for NDVI exposure. So we calculate the average exposure from 0 to 10, the early life exposure, which was modeled but basically corresponds to the exposure at around birth. And we also have a metric of the change in exposure through time. And as you can see from the estimates, increasing levels of NDVI over time are associated with a slight increase in the lung function parameters in these cohorts. The early life exposure was associated with these lung function parameters in the crude model, but after adjustment, these associations no longer subsisted. We also reached this null finding. So in this study, in this project, we found no evidence that blue spaces or proximity to urban green spaces was or play a role in the development of asthma or allergic diseases. We can again discuss this later on, but it's also important finding of the study, although it is not a good finding to sell in the paper. And we also reach another null finding or less conclusive finding, where we were not able to find any mediation effect of physical activity or NO2, so the traffic air pollutant we use in the associations between green and blue space and the 3 studied outcomes. So in the cohort, these were the only mediators we were able to assess. So these results seem to suggest that either the effect is direct or it is mediated by other variables, such as psychosocial variables related with stress that are not available in the cohort. Of course, these results and the project I present to you have several limitations that are worth mentioning. The first limitation is the fact that we do not have any direct measure of green space utilization. So we assume that having a urban green space near the residence is a good proxy of utilization, which we are aware this might not be true. We don't have any measure about the perceived neighborhood environment, which has been discussed by other researchers, can be as important as objective assessments. And many studies indeed were able to report that is more likely to find associations with perceived measures than with objective measures, because these perceived measures took into consideration the perceptions and in the experience of the participants. In these projects, we also use traditional definitions of neighborhood, so buffers and census blocks, which might not match with the activity spaces of the individuals. So in other words, this work, as many other works, is affected by the uncertain geographical context problem. Our data set on air pollution, as I mentioned, has a low spatial resolution, so this might have limited our capacity to explore the mediation effect of air pollution. The data set on biodiversity also only includes animal macro species, so we don't have species of trees or flowers or etc. So this is a big limitation, but it was the only data set available, and we didn't have money to make our own collection of species available, maybe in the near future. And again, as many other studies in the field, our results were sensitive to the chosen buffers and the chosen definition of neighborhood, which means it is quite difficult to translate these findings into urban planning interventions, because urban planners usually do not deal very well with uncertainty, so they want to have very concrete solutions for their problems. So just a little bit about future perspectives, because it might be interesting to you. So this project has ended in June, but we are starting other projects at the moment. We are starting this project called UG that aims to understand how the housing crisis and touristification in the city of Porto that led to gentrification and displacement are affecting the health of the population. And for that, we are going to use data from Generation 21 to have the young people, and Happy Porto to have the old people represented in the study. This study also will involve a series of qualitative studies. We are also at the moment within the scope of the cohorts evaluating COVID-19 seroprevalence or incidence of the disease, and also long forms of COVID-19 disease. And we intend to link these two outcomes with housing characteristics and also neighborhood characteristics. And within the scope of a series of theses, and within the scope of my own project that pays my salary, basically, we are also looking at the role of neighborhood environment on healthy aging using Epiport cohorts and other European cohorts. So just to end up, I just want to say that in Generation 21, we have quite a lot of information on the environments of these individuals, and also on the health of these individuals. And this information can be used to conduct environmental health studies and urban health studies, and it can be capable of generating evidence to guide policy and urban planning strategies, such as the evidence we generated through the EXALAR 21 project. So to end up, I just want to give a big thank you to all people involved in the Generation 21 cohort, both participants, families, and staff. I want to thank the members of the EXALAR project, because they were the ones developing most of the work I present to you. And I also want to thank you for your attention. So if you want to reach me, you can use my email address or other kind of social media.